am Sajini, working as an assistant professor in Department of Pharmaceutical Chemistry, Yenepoya Pharmacy College and Research Center, Yenepoya, deemed to be University at Mangalore. Today I am here to give the lecture on Introduction to the Molecular Docking. These are the table of content I have included in my slide, that is an introduction, molecular docking tool, general procedure, application and case studies, future trend of molecular docking and reference. Before proceeding to the molecular docking, let me give the introduction on what do you mean by CADD. CADD is nothing but computer-aided drug design. By use of computational technique or by using the software, we can design and discover the new drug molecule. From this, we can determine or we can predict how the new drug molecule can interact with the protein or enzyme, anything. From there, we can optimize their effectiveness and we can improve their safety profile. And before doing any in vitro or in vivo study, if we do the in silico study, we can save our time as well as the resource. So these are the type of in silico studies, that is a molecular docking, molecular dynamic simulation, quantum mechanics, pharmacophore modeling, QSAR and virtual screening. Molecular docking I will be explaining in the coming slide. Molecular dynamic simulation. It is a computer model and as name suggests, molecule in the sense it can be protein or the ligand. How they can be moved, how they can be changed over time, we can study by using molecular dynamic simulation. And from molecular dynamic simulation, we can study how they are fit together, how they can be interact with each other. From this, we can understand how they are functioning in our body, how they are behaving in our body. We can study by using molecular dynamic simulation without any physical experiment. Quantum mechanics. Quantum mechanics, it is a method. It will uses the science to understand how the atom or the molecule will behave in the detailed manner, how they are interacting with each other, how they are bonded with each other. We can understand by using the quantum mechanics. By seeing the structure of the molecule, we can determine how they are behaving in the different situation. So quantum mechanics will be the very helpful. Next will be the pharmacophore modeling. Pharmacophore modeling, from this we can understand what are the key features of the molecule is required for the binding to the target site. From this, we can design the new compound by keeping all these key features for designing the new molecule for the interacting with the target site. QSAR, quantitative structural active relationship. It will determine how our molecule will be relates to the effect of our body. Virtual screening. Virtual screening is nothing but it is searching of large molecule. From this, we are determining the one compound which are best fit between the target site. Here two types will be there, ligand based virtual screening and structure based virtual screening. Ligand based virtual screening where 3D structure of the protein will be unknown. Structure based virtual screening where the 3D structure of the protein will be known. So ligand based virtual screening in example will be the pharmacophore, QSAR we can perform. Structure based virtual screening we can perform like molecular docking simulation. Now we can come to our topic, that is a molecular docking. As name suggests, molecular and docking. Molecule means smallest unit of the compound. Docking means its process of docking or fitting the one molecule to the other. So if we are having many ligands, we are finding the best fit between the molecule with the target site. Target site can be anything. It can be protein or enzyme. So definition says as molecular docking is a computational technique used to predict the binding affinity or the proper orientation of the ligand molecule within the binding site. So what do you mean by binding affinity? Binding affinity is nothing but it will be determine the strength of interaction between the ligand and the target site. In simple term, we can tell that what is the strong bond between the ligand and the target site. If the binding affinity more means there will be a stronger bond between the ligand and the target site. If the binding affinity less means the less bond will be exist between the target and the binding site. So, this binding affinity will be very important in drug discovery process. From this we can determine if the larger the binding affinity means drug will be more effective in the low dose of the drug. So, if when ligand will be binded to the target site, there is a various force will be involved. It can be van der Waals, hydrogen, hydrophobic, ionic interaction. All these interaction make the system lower the energy. So, whatever the binding energy will get in the negative term. So, larger the negative value means more the binding affinity between the ligand and the target site. So, docking score is larger the negative means there is a stronger bond between the ligand and the target site. Next will be the proper orientation. Proper orientation means stereochemistry part, the 3D arrangement of atom within the molecule. If there is any small change in the 3D arrangement of atom means there will be impact to the binding of the ligand and the target site. So, many factors will be taken into the consideration. It can be the chirality, RS configuration, the cis and transform, or in stereospecific. If we consider the chirality, if molecules having the same composition, different arrangement, we call it as the enantiomer. One form of enantiomer can bind properly to the target site, but another form will not. Other form will it can bind, but it can provide the unintended effect or the side effect. So, proper orientation selection is very important in order to binding to the target site. If we consider the chirality, carbon is if the carbon will have the chiral compound, it can exist in the R or S form. So, only one form will be well fitted to the target site, other form may not. If we consider the double bond ligand, it can exist cis or transform based on the atom will be surrounded around the double bond. So, only one form will be more specific, other form do not. If we consider the stereospecific, many macromolecules which is available, it is stereospecific. Stereospecific means it will be bind to the only proper 3D structure of the ligand. If it bind to the other form, it will do not form the any kind of interaction. Interaction can be the hydrogen bond, hydrophobic, ionic interaction. Proper interaction may not be there. So, it will be while selecting, it should be proper orientation taken into consideration before binding to the target site. So, these are the type of molecular docking. It can be the rigid docking, flexible docking, induced fit docking, blind docking, targeted docking and fragment based docking. Rigid docking meaning it will be both the target as well as the ligands are rigid in nature. Flexible docking meaning only the ligand will be flexible in nature, so it can be generate all the conformation, whereas the protein will be in the rigid form. Induced fit docking meaning both the ligand as well as the protein are flexible in nature. Blind docking where the ligand can bind to any part of the protein. The predefined part was not be defined. So, after docking score, we can define the site where the we can determine the site where the binding was occurred. Target docking is nothing but it will be predefined the site on the protein where the ligand will go and bind to the selected site. Fragment based docking meaning if your ligand will be broken down into the several fragments and each fragment will go and dock to the protein. Laterwards, ligand will be grown and it will be binded together. So molecular docking tool, Schrodinger, Pyrex, Autodoc, Vina, Gold, MOE. If we consider the Schrodinger, it will offer the many software in the case of application of the pharmaceutics. Mastro is a control panel. It will assess all the tool which is helpful to the studying the molecules. Mastro software, it will use to predict the molecular docking, and Desmond, it will be the molecular dynamic simulation we can study, and Prime where homology modeling we can study, and the Quick Probe where pharmacokinetic of the ligand we can study. So next will be the general procedure of the molecular docking, that will be the protein preparation and ligand optimization. It will be the ligand preparation, receptor preparation and docking simulation. Initially ligand preparation, ligand can be drawn by using the ChemDraw or the ChemSketch software. Then protonation has been done. Protonation is nothing but it is a charge on the ligand. It is based on the gaining or losing of the proton, and protonation plays an important role when it binds to the protein site. So if for example, if the ligand is having the positive charge, mean it will go and bind to the negative part of the protein. So before docking, protonation of the ligand is a very important in order to get the by adjusting the pH of the environment we can protonate. Next will be the 3D optimization. 3D optimization of the geometry, meaning it comes two factor, that is a conformation generation, energy minimization. Conformation generation, ligand can adapt various shape and form. So if the ligand is a flexible, means it can rotate, it can bend, it can twist around the bond. So this flexibility allows the ligand to bind to the target site more properly. So when the ligand will be adapt multiple shape, they will be generating the many conformation, and all the conformation will go and dock to the protein site. So energy minimization, lower energy conformation has been selected. Lower energy conformation will be having the more stable form. Again, tautomer and isomer generation. Tautomer, if the ligand is having both keto and enol form. If the ligand is having the chiral center, it can be a chiral center in the sense it can have the R or the S configuration. Any one form can be go and bind to the target site. So before going to the docking procedure, it is important to select the any one form of the ligand. Next will be the receptor preparation. Receptor preparation, protein selection. Protein selection we can done by using the RCSP protein data bank, and based on the resolution, based on the biological relevance, based on the co-crystallized ligand, we can select our the protein in the PDB format. Once the protein is selected, then we can go to removal of unnecessary molecule. Water molecule will be there in the protein structure. It is not at all required for the binding, the docking simulation. It is there for the stabilizing the protein. So water molecule can be removed off. Next will be the co-crystallized ligand. Co-crystallized ligand, meaning there will be the bonded ligand will be there in the crystal form of the crystal structure of the protein. So it has been removed unless we are studying the competition between the two ligand with the same site. So iron and other protein, whatever there in the protein structure also removed. It is not at all required for the study of molecular docking. Next will be adding the amino acid residues. When we are downloading the protein in the PDB format, there will be the missing residue will be there. By using the software, we can fill all the missing loop of missing residue of the amino acid. Next will be the protonation and pH adjustment. protonation and pH adjustment is a very important in the receptor protein preparation, because it is required for the binding interaction. If protonation, meaning there is a charge of the amino acid in the protein, so its right charge is important for the interacting with the ligand for the strong interaction. If pH adjustment, we have to adjust the protein in the normal pH of our body, because it will be reflect to how it will be work in our body. And next will be the hydrogen bond adding. Hydrogen bond adding is required for the interaction. If it is not there, there will be no interaction between the ligand and the target site. Next will be the energy minimization. Energy minimization means there will be the unnatural strain protein will be there, which is not at all required, and it has been removed, and lower energy protein will be selected for the further studies. Last, conformation. If the protein is the flexible in nature, it will adopt many conformation. So based on our studies, we can select the relevant conformation. Based on our study, we can select. Next will be the grid generation. Grid generation is nothing but plotting the graph around the protein, and there is some spot will be generated where the ligand will go and bind to the specific spot. So the scientist can understand where the ligand will bind more properly and best position. We can understand. Once the receptor preparation is done, we can go for the docking simulation. So, scoring function and ranking algorithm. Scoring function is nothing but it is a mathematical equation, and it will be evaluate how our ligand will be bonded to the target site. If it is mainly based on the binding energy and the interaction, and the ranking algorithm is nothing but based on the score, it will be ranked the ligand from higher to the lower score. If the scoring, if important of the ranking algorithm will be best candidate will be chosen for the further studies. There are three type of scoring function will be there. That will be the force field, empirical scoring, and knowledge based scoring. Force field is nothing but it will give the score based on the physical force. Physical force is nothing but it can be the attraction, repulsion, ionic interaction between the atom of the ligand towards the protein. Empirical scoring, it is nothing but based on the mathematical equation, based on the factor like hydrogen bond, hydrophobic interaction, ionic interaction, all these it will add up and give the score. Knowledge based scoring is nothing but it will based on the prior knowledge, based on the database of known ligand and target bond. Based on that idea, we can predict the new compound, how it will be interacted with the target site. Based on that, score will be given. Now, application and case studies. During COVID-19 pandemic, the already existing drug, that is hydroxychloroquine and remdesivir molecular docking study was performed in order to see whether this already existing drug go and bind to the protein, that is SARS-CoV-2, in order to prevent the infection of the cell. Next will be the HIV protease inhibitor, that is a saquinavir. Saquinavir was designed by using the molecular docking method, and it will stop the enzyme, that is protease enzyme, and it will be used in the HIV cases. An antiviral drug, that is oseltamivir. Oseltamivir is a flu medication. It is used in neuroamidase inhibitor, and it is an antiviral drug. Not only that, if we consider the Alzheimer's disease, many potential drugs will be docked against the tau protein in order to see whether it will potentially decrease the progression of the disease. And imatinib, it is an anti-cancer drug, also designed by using the molecular docking method in order to see whether this drug can potentially inhibit the protein which is responsible for the cancer. Last slide, it's a future. What is the future trend of the molecular docking? If we apply the AI tool in the molecular docking, what are the advantage we'll get? That is a better prediction, automation, improve the scoring, fast screening and the safety prediction. Better prediction is nothing but if AI tool can read all the interaction between the ligand and the target site, it can predict what is the interaction between the new drug towards the target site. From this we can determine, the result will be the more accurate. Automation in the sense molecular docking process require many steps. So by using the AI tool, we can automate all the steps, so we can faster the result will be getting. Improve the scoring. By using the smarter scoring function, we can by using the smarter scoring function, by analyzing all the interaction between the ligand and the target site, we can select the more best candidate of the compound for the further studies. Fast screening. Fast screening is nothing but if AI tool can screen 1,000 and million the compound with a fraction of second, means our result will be more faster. Safety prediction. Safety prediction in the sense AI tool can predict the safety or the toxicity study, mean we can select the more safer drug for the drug discovery process. These are the reference. Thank you.